Welcome to ETSU Department of Psychiatry Grand Rounds. Dr. Hendrick is our speaker today. Dr. Hendrick is Chief of Psychiatry at Mountain Home VA Medical Center. He is an Associate Professor of Psychiatry at ETSU. He also directs the Neuroscience Didactics track for the residency here. A graduate of the Medical College of Virginia, where he was also trained in psychiatry. He is a Distinguished Fellow of the American Psychiatric Association, holds a Distinguished Service Award from the United States Public Health Service, recipient of the Irma J. Bland Award for Psychiatric Resident Teaching by the APA as well, and twice been named Psychiatric Resident Teacher of the Year here at ETSU. Dr. Hendrick's focus is on behavioral neurology and neuroanatomy as it applies to behavior. Today he's discussing how evolution has been the driving focus of the functional complexity of our individually personal 3-pound universe, the brain. Dr. Hendrick.
Well, I hope I'm not boring everybody being back again after such a brief time. We had a little glitch in the ground schedule, but fortunately this was already set up and we were able to shift things around. I started last time with a quote from Hippocrates that begins, "Life is short, art long." And what I'm going to say to add to it this time is mutants in genes, consistent, evolution, constant, and the changes always occurring. Now, they occur over long periods of time. I'm going to start with the financial disclosure, which is up on the screen. I don't have any disclosures except for my employers. However, I'm going to extend my disclosure in this case because there are two kind of contradictory and sometimes seen as conflictual things. I'm a devout Episcopalian, and I very much believe in that. I have faith in God, and this is something that I feel in my heart. Meanwhile, I very much believe in the theory of evolution, and those two things don't often go together. I believe they're quite segregated from one another, and I think people that don't see that are mistaken. I'm not a fan of Bishop Ussher, who believed that the world was created more or less in 1 week in 4004 BC. But then I'm also not a big believer in Richard Dawkins, who believes that there is no God and that atheism can be described to a large extent by the nature of the theory of evolution. So if you were in my grand rounds a little while back, you may know that part of the beginning of wisdom is to be able to take ambivalent ideas and put them together in some constructive way, and I'm going to try and do that a little bit today. But that's my disclosure of my personal biases in this. So, the universe began 13.7 billion years ago in a Big Bang. And during that Big Bang, many things happened. Basically, the nuclear formation of the universe occurred in a very brief period of time, leading over billions of years to the formation of galaxies. And as this happened in about 3.5 billion years ago, the beginnings of nucleotide synthesis, the beginnings of complex organic compounds that could have something to do with life, began to develop. Those compounds, of course, are subject to the typical thermodynamics of the reaction that you all remember from a general chemistry course. There's a heating period and an activation energy has to occur in order for materials to be produced. And over time, catalytics can change the dynamics of this equation markedly. But nonetheless, we have to start with what we might call non-living organisms. So what this means is these are very small chemicals capable of actually causing their own reformation. They aren't life per se, but they are reproducible objects, if you will. And we can experimentally demonstrate by putting certain acids and various bases, heating them under pressure in condenser tubes, we can demonstrate that eventually what you will do is form basic sugars, basic organic chemicals, and the basic building blocks of life. And so from there, then, living organisms somehow evolve, and they come from a variety of different possible sources. For instance, the heated water range around underwater volcanoes could be one source. Another interesting idea is that clay, which you may know forms sheets that slide against each other. These sheets have various degrees of adhesive capacity, depending on the moisture around them. And it may be that what happens is these non-living chemicals could bind in differential ways to the sheets of clay, leading then to the ability to construct ultimately a bipolar membrane, which is probably the necessary key for the initiation of anything that we would call a living organism. So there are actually several missing links. One is, how did we go from either underwater volcanoes, or how did we go from sheets of clay that bound molecules? How did that happen and go from non-living to living organisms? And of course, this explains it all, I think. So we would like to be a little more explicit about step 2, but we're a little dumbfounded. That's one of the missing links that we don't know yet. But we do know that ultimately, evolution led to RNA, and from RNA, there was a process in which proto-organisms utilizing RNA began to develop a mechanism to replicate in such a way that amino acids could become associated. And with the introduction of DNA into this process, eventually we got to the place where we could structurally define proteins. And with the definition of proteins, animals were able to go from being nothing more than simple flatworms to much more interesting and larger encephalized animals, both on land and in water. There were multiple mass extinction events during the emergence of various forms of life. But even as far back as 540 million years ago, and you'll see in this trilobite, you'll see that the process of encephalization has been critical in understanding the nature of evolution. So here's a very ugly looking little worm, or maybe beautiful if you want to look at it that way. And it's thought that the DNA content of this worm may very well be sufficient to build a human brain. Now, it won't do it. The proper chemical environments and the proper biological keys are not in play. You can't actually do that, but it may be that the DNA content actually has the programmability. It's just that the biological nature of that programmability is unavailable. So we come now to another missing link, the Cambrian Explosion, or the Cambrian Period. And in the lifeline of animals, we came to this point in time where we had many flatworms and things of that nature, sponges, those kinds of things. But then in the Cambrian Explosion, all of a sudden, multicellular eukaryotic organisms that became much more complex, that were quite a bit more encephalized, that had at least early neural systems, all of a sudden this huge explosion of new animals evolves. And again, we're not sure why that happened, but it seemed to. And so here's a fossil of the trilobite that I mentioned, and you can see how encephalized this organism is. So this process of moving into complex animals with encephalization has been a cornerstone to understanding the nature of evolution. And the encephalization, of course, is to accommodate primitive brain. Now, this is reptilian brain, and it has certain markers of things that we're used to seeing in mammalian brain. There's a primordial cerebellum. There's a red nucleus, and some of you, those of you that have taken my class, know the red nucleus has a lot to do with the crawling reflex. So in an organism like this, that obviously can't stand but must crawl and shimmy its way along, the red nucleus is a driver. You see the typical medulla, the pontine area, the pontine cerebellar area, the midbrain tegmentum, and then you see a primitive thalamus for sensory gating, and you see what is referred to here as the DVR on top of the stratum. The DVR here is basically a dorsoventral reticulum. So this is not cortex with a prefrontal area and executive functions. This is a driver zone that drives through the basal ganglia, called here the striatum, into the red nucleus and other regions down to the motor neurons of the limbs, so that in fact these older organisms can crawl along the ground. And moving from this kind of early encephalization into more human-like beings, we see that humans went down many various cul-de-sacs on the way to becoming Homo sapiens. As a matter of fact, Neanderthals were successful enough from about 130,000 years ago that we still see a small percentage of Neanderthal DNA embedded in the human genome, which is an interesting crossbreed. But by and large, Neanderthals did not have sophisticated enough telencephalons to be able to continue. And about 250,000 years ago, Homo sapiens appeared on Earth, though they did not reach modern form for another 200,000 years, becoming known 50,000 years ago as Homo sapiens sapiens, a term which means those that know that they know. And what do we know? Well, what we know is we have foreknowledge of our own death, and it is not clear that Homo sapiens before about 50,000 years ago were clear on the fact that ultimately there would be a terminal event in each individual's life. So when we look at the brain, we notice some things that are the same all the time. The cortex has 6 layers. The self-free zone is an associational area. Then there are cortico-to-cortico translational and connecting layers. There's a thalamic somatosensory gating area. Area 5 typically is the transition in the subcortical areas, and then area 6 is known as the thalamic outbox because the thalamus does have bidirectional connection to the cortex. It's organized in columns. It can be divided into roughly 20 specialized areas, maybe a little bit more in the human. The primary areas are surrounded by areas that have secondary and tertiary focusing properties for a sensory or motor system. Visual is in front. Visual area is processed in the back. Somatosensory is in front, and auditory is in between. Now, in different animals, we see this process of evolution developing at different paces. Though you'll notice here, the largest climb apace is that of the human from roughly 50,000, maybe even 250,000 years ago. But interestingly, in this slide, you'll note something else. Note the dolphin, okay? It's on kind of a different glide path, but the dolphin, too, is a fascinating animal and has its own specialization for its life in the water, as do humans terrestrially. And in that regard, it's because of their encephalizations of the 2 species, okay? Basically, the gross brain size does represent the neuronal processing capacity, but it's not necessarily related to body size. Also, in addition to that, the frontally placed eyes are very important. For instance, in humans, they're very important because they're part of our predatory history. Prey animals have eyes on the sides of their head. So horses, for instance, have their eyes on the side of their heads, whereas wolves have their eyes in the middle. And I have an entire story that my behavioral neuro residents have heard about that, but I'm not going to bore you with it today. But this placement of the eyes has a world of things to do with how an animal fits into its environment. That also is associated with this precise control of the musculature, and this allows for increasing complexity of social environment, no matter what species you are, and that allows for experimentation with change in diet, which is very important in the process of evolution. So when we get out of the reptilian brain and up to the mammalian brain, we see more things. Now, there are more things similar here than dissimilar, but nonetheless, we see some new and interesting things. For instance, the amygdala becomes more important. So watching for fear, threat, and for opportunity, okay? Greater sense of olfactory input, The striatum is now actually basal ganglia instead of just a routing device. The thalamus continues to be a major somatosensory portion, but we again see the medullary, pontocerebellar, midbrain, diencephalic, and telencephalic regions, part of the forebrain. And these are all the things that we typically see in mammals. Now back to our dolphin. Some interesting things here. This dolphin has a very, very symmetrical brain. Its sulci and gyri are symmetrical. The sizes are symmetrical. The cerebellum is larger than per capita in the human brain. And why is this? Because in dolphin evolution, it's very important for dolphins to be able to avoid attack from above, below, right, left, anterior, posterior, in all regions. They have to be equally adept. Otherwise, if they were left-handed, say, they could constantly be attacked on the right and lose. So the dolphin brain is very symmetrical and properly adapted through evolution for the aquatic environment, whereas the human brain benefits greatly by being able to develop in such a way that it has lateralization. Now, in the lower part of this slide, we see another major feature. As we go down the phylogenetic scale, all the way down as far as the rat, we see increasing lysencephaly, or smooth brain. Smooth brain is not good. That outer layer of associational zone, what was called in the cortex the cell-free zone, is tremendously enhanced in surface area by the gyri and sulci of the brain. And that associational area, the degree and size of the telencephalon, the amount of associational neuron connections, is tremendously influential in what makes us capable and smart. Rats have a very limited behavioral repertoire. They're tricky, but they are not really smart. The brain, of course, along with the peripheral nervous system and the skin, comes from the ectodermal layer. And the way this happens is cell migration occurs over long-distance migrations, initially along glia, but then tangentially to various portions of the brain. Now, I wasn't able to get my Boyd's video to work, and so I don't have it, but if anybody wants to, you can Google Boyd's, B-O-I-D-S, and you'll be able to see these three separation, alignment, and cohesion factors at work. All of you have seen starlings gathered on telephone wires, especially during migration season. If you go up to Princeton Road and Roan Street on those telephone wires, there's typically about 40,000 starlings there in the morning. And if they're startled by something and they go, okay, you'll see these phenomenon in effect in nature. There's a cohesive movement, but there's a steerage, and there's an alignment of each one of the birds to another. And even 40,000 of them can jump off all at the same time and manage to go into these amazing swirls that birds are capable of. And the same general principle applies in cell migration. So certain neuroblasts move into these regions, the rhombencephalon, the mesencephalon, and then build the brain, which is twisting, gyrating, and sulsating at the time, in order to give us these complex interwoven neuronal networks. So as you can imagine, cohesion, alignment, and steerage is very necessary to make something this complicated organized. But when we look at it a little more closely, it is amazing how closely organized it actually is. It is truly an interwoven grid. It's almost like somebody did it with knitting needles. And that is surprising. How does this happen? Well, through neurogenesis. Stem cells start off in the neural tube, the ventricular zone. Ultimately, that neural tube ends up with stem cells in the subependymal region of the ventricles of the brain. There are induction signals. Various mitoses occur until neuroblasts get to the place that they can no longer divide. So as neurons eventually become fully differentiated, they can't be reproduced. But stem cells can reproduce at certain levels. So can we replace hippocampal cells? Probably. Can we replace corticospinal cells? No. Too differentiated. So this neuronal migration is guided by adhesion molecules in the peripheral nervous system. Okay? And then there is this differentiation that is cell autonomous and genetically described. Induction signals occur, critical periods for migrations occur. All of this leads to the construction of circuits in the brain. Trophic chemicals and attractants, repulsions, semaphorins are used to tell certain cells where to go. These neurons are linked in different regions, and these linkages, not only by white matter, but by something I'm going to come back to and talk to you about, bipolar neurons, are very interesting. And this happens by these various cell-to-cell signaling factors in which chemical cues and growth cones occur. There are decision points that cells have to reach. For instance, does a cell cross the midline in a decussation or not? That's a very important distinction for cell. And then ultimately, synaptogenesis begins. And synaptogenesis and its associated process, apoptosis, begin to develop things. Now, we see interesting connections here. For instance, superior cervical ganglion. Those of you that are thinking about neuroscience things, we get into the question of Horner syndrome and why does it involve the superior cervical ganglion? Well, there's an embryologic reason for it. And the same is true with blood vessels and the ear. So as axons grow out and reach their targets, they retract or regrow synapses. The more information flow that is affected at any given synaptic intersection causes that synapse to become either stronger or weaker. And then neurotrophic factors such as cahedrins are used there to long-term potentiate effective neuronal connection. So cell-to-cell signals, nerve growth factor are all a part of this. And apoptosis, what happens here? This is not the same as necrosis. This is disassembly of cells. And so, for instance, especially in very young children, in infant children, as many as 80% of cells can be just disassembled. And what happens is they are disassembled because there's a lack of neurotrophic factors in a region, or they've made a wrong or a non-connection. And if they do that, then that connection will be giving odd signals and it won't reinforce the cell. If the cell doesn't get that informational reinforcement, it'll tend to go away. So synaptic rearrangement occurs by competition. There's formation of new synapses when old ones are eliminated. Neurotransmitter release has a lot to do with being in the right place at the right time and causes this to be an activity-dependent phenomenon. And when a losing axon retracts, remember, Mother Nature abhors a vacuum, so a new, more informationally organized neuron will enter that area. Okay, so now that we have an idea how the brain builds itself, when does consciousness arise? Well, you see here about 500 million years ago, the Cambrian Explosion, but then it's not until Homo sapiens at about 250,000 years are around, even then another 150,000 years goes by before significant language begins to develop. Neanderthals probably had little more than a series of grunts that might be considered as emotionally differentiated. But actual language, and the language of things that have to do with expressing emotion, even more primitive emotions like anger or satisfaction, probably began to occur somewhere between 100,000 and 50,000 years ago. But one of the reasons that Homo sapiens become important is because not only do they begin to develop a knowledge of their own fore-death, but they also begin to use tools, and they begin to use tools quite effectively. So as this occurs, our civilization takes another large step in evolution forwards. And in terms of these consciousness, and I'm going to come to some of the specific features of Homo sapiens sapiens consciousness in a bit, Dennett has this idea. There is the plenum, that is the area in which your perceptual awareness exists. And from that plenum, there's an interaction with sensory input that then goes through a series of things in the brain. Now, in this particular simple drawing, we can call this the optic chiasm and the superior colliculus, the occipital region, and the temporal zones. So maybe I went through that a little quick. Chiasm, superior colliculus, occipital region, and here's the temporal portion of the visual system. And all of these things occur. Now, this is a very simple drawing, and I want to point out that as sensation progresses through these regions, it's not this simple. It goes through many, many hidden layers, as you can imagine from that complex drawing I showed you of the neuronal wiring. And these hidden layers have many implications for not only consciousness, but also evolution. Paul MacLean brought us this very interesting idea that in the hindbrain, there's reptilian function, okay, largely working around the red nucleus and the motor system, cerebellum. Then there is a limbic system in mammals, depending on what mammal you're talking about, larger or smaller. And then there is a neo-mammalian system, the cortex, which is actually a large inhibitory mantle. So emotional display can occur, but emotional display is not always good on social grounds or evolutionary grounds. So the cortical mantle actually inhibits inappropriate emotional display. So in this way, there is a dorsal ventral organization of the brain that has evolved, but that's not its only level of evolutionary organization. There is also a right-left organization, not well seen in dolphins. It's very symmetrical there, but in humans, it's very well seen, with the verbal apparatus located on the left side. And the verbal apparatus is a good example of the anterior-posterior evolution of the brain as well. We see here the arcuate fasciculus passing from Wernicke's to Broca's, giving us an idea of one of the white matter information superhighways that exists in the human brain. So these three different levels of organization, dorsal ventral, left, right, and anterior, posterior, are very important in the way the brain sets up. And that has been true throughout evolution, but the complexity of the brain has little to say, I said this wrong. The brain's actual complexity is not well defined by its size, shape, and fissures. It is well defined by total volume, okay? That's part of it, much better than here. But in spite of that fact, it isn't really the total volume. It is the area of association neurons, the total number of association synaptic connections in the cortex that make a difference. Nonetheless, the overall size of the brain does matter. And so we have our friends the dinosaurs talking about us behind our backs. But the real feature is the associational area. And you see here, compared to even monkey, a close relative in the primate group, you see how much of the human brain is dedicated to associational areas. Now, underneath this cortex is a very complicated subcortical wiring. And you see here, this is a non-human primate, okay, but you see the complexity of these hidden layers of wiring. And I want to make the point, again, we can't yet trace all these hidden layers. We can in various regions. We can do it more so in animals that are easily sacrificed, so we don't do it in humans as easily. But we have learned from these kinds of things that mammals are capable of general learning, but primates have specific social intelligence. And then when we get to the level of the great apes, relatively sedentary, but very structured in their social context, they have theory of mind. They can understand the beliefs of others of their species. Man has this capacity as well, though mankind also has the added feature a very sophisticated language function due to that left-right organization, anterior-posterior organization. So primates around 60, 70 million years ago, anthropoids about 35 million, and you see us devolving into our current back-breaking, index finger pointing, computer-generated postures. The first humans split from apes about 7 million years ago. Lucy, the mother of us all, about 4 million years. We went through a variety of attempts at Homo something till we got to Homo sapiens. And Neanderthals also coexisted with us for a period of time. But modern Homo sapiens, around 50, maybe 35,000 years ago, depends on who you ask. So, how did we get to our current modern man status where we're all worried about charisma, image, and sarcasm? In terms of charisma, I want to thank everybody in the room that has red on because you're thinking about Women's Cardiac Health Day. That's why I have my red jacket on today. So, that's a good thing. And I'm only being slightly sarcastic about that. How did we get there? Well, the size of brain parts is predictable. Okay? Neurogenesis has a lot to do with generating such things as the telencephalon. And the telencephalon, as you saw from the previous slide, and its association areas are huge associations. The laminar differentiation, for instance, the 6 lamina of the cortex, lamina 4, the thalamic inbox, lamina 6, the thalamic outbox, and the cingulate gyrus, which is the cortical level of control of the limbic system. There are also 6 layers, but the 4th and the 6th layer are not differentiated for thalamic input and output. The 6 layers have similarity of numbers of cells. And then as we evolve, we get increased circuitry for biophysical senses, and we get vision, audition, and sensation, very important in the brain. And then in addition to that, as the brain evolves, there's an increased diversity of cell types. So for instance, we see things like stellate cells and basket cells, even more so than pyramidal cells, which are there for motor purposes. Now, there is this misconception that ontogeny recapitulates phylogeny. What does that mean? It's an old biological saying. The idea is that as animals develop from their embryonic state, they look a lot like they had in their evolutionary phylogenetic history. So a frog embryo on day 1 can look a lot like a human embryo on day 1. But the reason that this is something of a misconception is frog embryos do not have the same potential at all as human embryos. They're different in that regard. So Dr. Goodkin was kind enough to reintroduce me to this term clades. And the cladistics include the idea that closely related species are likely to share more biological features or characters. And while that's true, it's part of why it's a misconception that ontogeny is really not just a recapitulation of phylogeny. So why are we smart? Why do we have complex and maintainable social relationships? Monkeys can have complex social environments, but they don't gain cognitive capacity as a result of it. Okay? And part of it is about diet. Now, chimps eat a wide variety of foods. Gorillas, who I told you have theory of mind, have a very simple diet. So it's not just diet. But it has to do with the complexity of the diet. Not only in finding the food, but in how it's extracted. So are there behavioral strategies to search for high-quality food? What is high-quality food? Are tools used to produce food? Do we remember where food is and what to do with it? How to make it work for us? Can we recognize it readily? And how do we teach each other about it? So humans were better at this kind of behavior than any of its competitor species, and as a result, came out ahead. So monkeys, for instance, have different strategies, and it leads to different digestive systems. So here's the interrelationship of natural phenomenon of feeding to the change in different digestive systems and different digestive strategies in monkeys. But what made the difference for people? How come humans got so far ahead? Well, guess what? And Shambhavi, I hate to point it out to you, it wasn't vegetarianism. It was the use of meat eating. Meat eating was critical at a certain time in human history because it made for an efficiency of digestive capacity that led to the integration of enough calories that those calories could be sophisticatedly deployed to develop this neocortical bark, which is critical to the nature of the brain itself. So again, there are the 6 layers. The first diverse neuron types are there. The expansion of the cortex leads to the complex sulsation and gyration that leads us away from lissencephalic brains. I'm not aware that there are any Neanderthal brains around, but I would be willing to bet you that they were very smooth compared to Homo sapiens. This allows to interconnected regions that form gyri, and this allows to interconnections by way of fissures that would otherwise not have occurred. Somehow my slide blanked out. Okay. Well, and as this happens, you see that the encephalization quotient, measured largely by the distance from lysencephaly, goes up tremendously in Homo sapiens compared to others. And when we look at this quantitatively, we see the neocortex in human becoming just huge in its size and in its association capacity. Okay, so as these brains have gotten to be Homo sapiens brains, not yet Homo sapiens sapiens, nonetheless, humans get the wanderlust, and they leave East Africa for the Caucasus in Russia, and from there spread out into Europe and Asia. So, even before Homo sapiens got to that subspecies Homo sapiens sapiens, these migrations were occurring from the African exodus. And why? Because even as language just begins to evolve, this is something that makes humans different. We have language. We can use it effectively. We're self-conscious, including conscious of the fact that we can die and that we won't come back once we have. We have the ability to teach one another. And that is not a clear ability all the time. If you look at monkeys, you know, can one monkey teach another monkey how to take a stick and poke an anthill? Yeah, they can do that. But can they do more sophisticated teaching than that? Can they do teaching about what do you do or don't you do in order to stay alive? Well, humans can. And in addition, along with our cousins, the gorillas, we have this theory of mind, which means we're able to understand how our behavior can then be used to cause changes in the behavior of others. And we do this by learning to attribute belief to others and understanding what their beliefs are. Now, how do we get so good at all these things? Because our consciousness continues to depend on this complicated series of interneuronal connections we have at the associational levels of the cortex, and even at the perceptual regions and the subcortical regions. These hidden layers do other things for us. They cause a variety of pacemakers in the brain that cause the brain to be constantly desynchronized. Many of you know that I do ECT. Well, what's my goal in ECT? I give an electrical charge to synchronize all the neurons, and they spike. Okay? I tell my cardiologist friends they like their regular spiking waves. My organ, the brain, doesn't like to spike. I have to hit it with electricity to make it spike. But the way we do that is so that we can briefly put the pacemakers quiet, and then the pacemakers, having been insulted into quiet, come back online, and when they do, allostatic mechanisms of repair seem to be occurring in the brain. So this layer of hidden neurons, even though we can't map them yet, we know they're very critical to understanding full brain function. Well, not only that, Humans have other things. We have a more complex psychological life, more than just theory of mind. For instance, we have narcissism. Okay? Now, is narcissism good for us? Well, you know, it worked out for David pretty well. David was able to put a big hurt on Goliath because he was very self-confident and, in fact, came out ahead. So, how do things like this happen more in humans than in others? Well, because, having a couple of interesting slide moments, because we have these very interesting neurons, bipolar or von Economo neurons, and they have this very interesting capacity. They act almost like a white matter tract, a large white matter tract, but they're single neurons, and they go from one single neuronal hub to another neuronal hub, specifically in the brain, so that certain information can transition from one place to another in a very brief period of time, so that, for instance, David, slinging that little rock in his sling, knew exactly when to let it go, okay? And he did it at just the right time, and there goes Goliath. The bigger they are, the harder they fall. And so these von Economo neurons are a very important feature of the specific capacities that humans have. Now, look at some of our other animal colleagues on the Earth. Well, who else has it? Look there, dolphins have a lot of them, okay? And whales. But in addition to that, elephants. Now, elephants are a little different. Elephants have larger brains than humans by quite a bit. Of course, their bodily size is larger. They probably don't have as many associational neurons in cortical layer 1, but they do have, many of you will recall from the various stories you've heard, remarkable memory. And the reason they do is because apparently their von Economo neurons are much more specialized for memory control than is the case with humans. So, kind of reviewing a little bit on evolution, we have RNA, then we get prokaryotic DNA, and we eventually get a eukaryotic nucleus. This evolves into multicellular animals that are capable of various sensory activities, developing then into manipulative behavioral skill, and then eventually moving beyond the reptilian into the stages that we can think of as linguistic, reflective, and philosophical. And that brings us, of course, then back to the mammalian brain. Now, when we look at this mammalian brain, we see some similar things. The myelencephalon, the metencephalon with the pons, the midbrain, okay? Here's our thalamus. Now we have a much more sophisticated hypothalamus associated. You notice we have auditory and visual integration here, olfactory. We have differentiated basal ganglia, and we have input from frontal, parietal, temporal, somewhere up there is occipital. I can't quite see it from here. So we have more sophisticated input, and this is what the evolved mammalian brain is patterned like and has been for billions of years. So, in this process, we have a behavioral output system, not terribly different from that of a reptile. We have a visual input system that is much more sophisticated than reptiles, okay, in mammals. And the olfactory selection system has a lot to do, amongst other things, it has a lot to do with valuation judgments of fear and threat that the amygdala places on behavioral decisions that we may need to make. So one of the comments that I'll make to the medical students when I give some of this lecture is, if you walk onto a battlefield and there are 5,000 corpses lying there, the smell is horrendous. Makes you not want to go there. On the other hand, your left amygdala is saying to you, but if I do, there's a lot of bounty that I can collect for free. But the right side of your amygdala is saying, yeah, but there may still be some warriors alive around here someplace, and I could die trying to get that free bounty. So this olfactory selection system and its input to the amygdala gets very involved in the evolution of human behavior. The hippocampal pathway helps us because it produces memory and pushes it into executive prefrontal areas to help make decisions about what's the best move. The amygdala is greedy and scared, but executive decision making is what's the best thing to do. Once a decision is made, the cortex feeds information into the striatum, or what's also known as the basal ganglia, which is getting information from the thalamus, the somatosensory gating area, and driving that down through the midbrain and the lower brainstem for movement. In addition, in terms of that philosophical understanding of ourselves, we have a body image. And this body image has to do with somatosensory input into the thalamus, that information then being relayed to the somatosensory cortex of the parietal lobe, and that information then being used to make premotor planning and motor responses that have much to do with our body image, including such things as where's our body located in space and material of that nature. Also, well, the local image, this is a little more complicated. The local image includes things, for instance, like the definition of where your hand and the various aspects of your hand are in space. Much of the parietal lobe is involved with helping you to understand exactly where your arm and hand positions are in space. And then memory I've already mentioned, but memory has also a lot of input not only into decision making, also into valuation that the cortex places on various judgments. So here's the overt behavior generator. You see the cortex generating behavior. It's being modified emotionally and mechanically by subcortical regions and then drifting down into the hindbrain. And then covert behavior, unconscious behavior, is constantly working in reapprehentation pathways, involving itself in our decisions in ways that we are not often aware. So the somatosensory animal behavioral guidance system is involved in helping us select goals and produce behavioral outputs that will achieve those goals. We have a goal selection system that includes our memory and various and sundry representations of the body and various and sundry representations of the sensory input systems. Then we have the behavioral guidance system. Takes into account bodily condition, what's our locomotion capacity, what is the sensory input, how do we perceive that sensory input. My wife and I had a chance to rewatch Rain Man yesterday, and Dustin Hoffman's portrayal of an autistic person and his deranged sensory input is quite amazing. We take it for granted that our perceptions work the same, but they don't necessarily. So how do we choose to act? All of these things come together. The actions we produce are the result of all of these factors that have gone into the nature of evolution pressing the brain forwards. And so here we see the cortex in its various and sundry regions pushing into a very well differentiated basal ganglia, using subcortical regions, including things like the ansel lenticularis, the region of the globus pallidus, very old regions. Thalamus producing somatosensory information to help us with these goal-directed behaviors, moving that into the cerebellum so that we can properly time the nature of these activities, and then moving down into the spinal cord for output. An important feature of this is to remember the older a region in brain is evolutionarily, the more ventral it is and the more medial it is. So for instance, right here at the level of the ansel lenticularis at the globus pallidus, the bottom third of the globus pallidus, known as the ventral pallidum, is the oldest emotional processing system of the brain, much older than any limbic region. So even when we talk about things like amphibians, there is a certain degree of emotional processing because of the presence of the ventral pallidum. Now, what is that? Well, it's kind of like, oh, that thing's on fire. I need to not go near it. That's kind of the emotional processing we're talking about at the amphibian level. But nonetheless, there's something there. So ultimately, we get to a manipulative behavioral animal guidance system. And this guidance system has been evolved. Those that effectively produce these guidances inside themselves tend to live, tend to reproduce, and tend to ultimately have mutations that go on to produce further evolution in the species, in all species. So our brains then, for a long time, look like this. There's a forebrain, a clear midbrain, there's a subcortical region here, and then there's the met and myelencephalon, there's a cerebellum, and all of these portions of the brain are necessary. And ultimately, in humans, for instance, we get to see imagination. Okay, this is a very high functioning level of brain, and probably true imagination may be something that exists maybe only in humans, possibly a little bit in gorillas, but not in a very sophisticated way. Does it exist in dolphins? I have no idea how to guess. But it's very interesting. And fortunately, even for all these millions of years and all these very complicated ways, we eventually did get to the evolution that we all wish to achieve, which is Homer himself. And with that, thank you. Questions?
Well, John, you covered quite a bit of territory there. One thought I had in response to the lecture was about neurogenesis in the adult brain. And we know it occurs in the olfactory bulb and in the hippocampus. And in the hippocampus has been related to major depression with decreased neurogenesis in major depression, and then with treatment increased. I wonder if you might want to comment on the functional implications of neurogenesis in the adult brain.
Yeah, actually, I appreciate that question. I do. There was information produced that the hippocampus was reduced in volume in studies in depression, and that it was then able to be regenerated to its previous position. I believe there were like 4 papers showing this, and then later questions about the statistical validity of that came into play, and it became clear whether that was true or not. After that, though, further look has shown that, in fact, antidepressants probably do that, maybe not quite as robustly as was originally alleged. But one of the fascinating things is there's also now been studies showing, I don't believe replicated yet, that ECT also does this. But not only does ECT restore olfactory and hippocampal areas, which one might imagine that it would, a region that I would not have suspected because I would have thought they were too differentiated, superior temporal sulcus also seems to regain size with ECT, but not with antidepressants. So it is a fascinating question to begin to endeavor to find out what is it that allows stem cells to reproduce certain cells. What are the factors involved in that? And I think it's just fascinating that that occurs, and how and why it occurs, I wish we knew.
Actually, just to give a step more on the side of interest that I have in that related to pro-inflammatory cytokines, cortisol has been associated with decreased neurogenesis in hippocampus, and also increased pro-inflammatory cytokine secretion, TNF-alpha, IL-1, IL-6. And those are increased in MDD. And with treatment of MDD, you see the counter-regulatory cytokines increase in IL-10. And also, this has been, neurogenesis specifically, has been related to BDNF.
Mm-hmm. Yeah, I've been concerned for some time that chronic use of cannabis may very well be turning down or even off BDNF. Worry about that because we've got a lot of veterans that are kind of hunkered down up in the hills smoking dope all day long. They've been doing so for 25 years, and I'm not sure it's done them any good on their chronic PTSD. And the data is out on that as well, but I am a little concerned that one of the problems with cannabis is the decrease in production of BDNF. It may have something to do with the reason that those that are potentially prone to psychosis are more likely to become psychotic if smoking dope is younger adolescents. Yeah, I think I didn't talk about that, but I had a lot of material in this talk, but I think BDNF is a very important aspect of all of this.
I'm going to shift gears a little bit. I just got there's a fairly growing literature on the similarity of psychotherapy to medications in treatment of depression. And now there's been 500 meta-analyses that show that. And so, you know, we're beginning to have some idea about how the medications affect the brain, the neurocognitive systems. How do you think psychotherapy might be, because it does seem to have a similar regulatory effect, how might we begin to conceptualize that from brain evolution
Well, I
perspective?
think one of the things that's going on with this kind of thinking is it's led to a tremendous amount of neuroimaging, and neuroimaging is supporting this idea over and over again. And I think one of the ways to look at it is, I was referring to synaptogenesis and synapses constantly being groomed, and they are. And I think that you don't have to, Carl and I are talking about actually growing new neurons. You don't actually have to do that. You can grow new synaptic connections. And the better informational transfer that occurs at any given synaptic region, the more likely you are to get long-term potentiation of those synaptic areas. Neurons crave information transfer. And so if you can reprogram, if you will, those synaptic connections in such a way through the use of psychotherapeutic intervention, easy for me to say, then I think that's the kind of thing that we can anticipate happening.
So you're thinking it's information. How about some of the idea of neuroregulation?
Makes sense. I mean, I understand what you're saying. I don't know that I have any data to address to that. But I don't think that there's any question that when we talk about neurons craving information, part of that has to do with craving positive, healthy social interaction. So that does make sense. Okay, thank you very much.